with introductions. So, well, thank you very much, everyone, for joining this this webinar. By the way, you see my screen, yeah? Just in case. Yeah. Good.
Yes.
No. Thank you very much, everyone who joined our today High-Affinity Talk. So really great to host today Eva Smorodina with a very, very interesting topic and very attractive talk about the structural modeling and the ability of AI tools to predict antibody-antigen pairing. Yeah. I'm Stan and I'm CEO of my laboratories. Just 1 minute intro who we are, why we post those webinars. So our company, My Labs, comes from academia. We are the creators of MyTR, and I already see some of our very good users joining this webinar, a tool for VDJ analysis, which is widely used in academia. And today we work a lot in the antibody discovery space. We are developing some software for antibody discovery end-to-end, from NGS to lead selection. And the topic of affinity modeling, structural predictions is extremely interesting today in the industry. That's what more and more companies, more and more people are doing. So that's why, Eva, now I'm stopping to share. Your presentation is so interesting to everyone. We'll have a presentation for 20-25 minutes probably, and then we'll have time for Q&A session. So there is a Q&A button in Zoom where you can post your questions at any time, and then we'll ask them.
Perfect. Thank you very much for such a nice introduction and for inviting me. I will have a talk about structural plausibility without binding specificity. And in simpler terms, it's about the latest AI-based structural prediction tools and how really practical they are in the drug discovery pipelines. But before diving deeper into that specific topic, I want to paint a bigger picture why we actually need that all. Computational antibody design would revolutionize the drug discovery because we will be able to screen more candidates in a more efficient way and reduce the time and resources needed for wet lab experimentation by providing better candidates just through computational pipeline. However, right now it is still a dream and not reality because we do not understand the rules of antibody-antigen binding. I personally believe that if you do not understand something, you cannot create it. So in this way, we do not understand the antibody-antigen binding, so we cannot create really great antibody and using just in silico method. Specifically, the antibody-antigen binding happen in 3D, and it requires both sequence and structure-based rules. With sequence, it's a bit more clear, and there are more information, more data. So I would say that there are already existing methods that are able to separate, let's say, high and low affinity binders just based on sequence information. However, that is not true for the structural, because there are less structures in sequences. The quality of structures for many antibodies isn't that good. And also there are many repetitive information and not sufficient information for the AI model. Due to all these limitations, in many practical cases, you cannot just use experimental structures, but you need to go from sequence to in silico modeling of the structure to function. And this in silico modeling step becomes crucial to get the relevant and realistic information about the function. If this intermediate step do not provide you with accurate structure, and by accurate, I mean the structure that represent the biophysical reality, then we also cannot get the realistic function in terms of the structural parameter. So answering the previous issue regarding limited understanding of antibody-antigen binding rules, we need to add more structural information and be able to understand better antibody structures to really solve the computational antibody design. This work and this talk is about one of the missing parts, and specifically how to accurately model antibodies and antibody-antigen complexes in high throughput. More specifically, It is the recent preprint that's called internally Antibody-antigen plus, thanks to a very famous anime that means a mix of different things. The main question of this preprint was to figure out whether structure prediction tools, and specifically AI-based structure prediction tools, can pick the right epitope-paratope pairs and detect the right epitope for a given paratope. More specific question is, can structure prediction tool differentiate the right and wrong paratope-epitope interactions? I assume that you all understand what is it, paratope and epitope. If you don't, please interrupt me and I can explain it. But let's say these are the binding sites, and the main approach and the main setup was to get the experimental structures of antibody-antigen complexes. We subselected the dataset of 106 experimental antibody-antigen complexes. Then we just took the sequences and repredicted those structures in a very different set. The sequences that we took, we tried to keep as different and diverse as possible to be able to track different parameters. For example, we tried to use PDB IDs from different ages, like some are older, some are newer. We also tried to keep different resolution of the experimental structures. We tried to keep different components of amino acid sequences and compositions and similar things. But now, let's talk a bit more about the structural reprediction that I mentioned before. So we took this 106 VHH and antigen complexes and extracted sequences from them. The first case scenario is the original experimental complexes. They were just from PDB, and there is nothing more to say about it. But when we come to reprediction based on the sequences one setup, is when we use absolutely the same pairs as were in the PDB and just provided to the structure predictor tools the sequences to get the same complexes. Such case we call real complexes. Another case was when we took the VHH from one pair and combined it with antigens from all other pairs. So in this way, we created negative data on non-cognate antibody-antigen pairs and repredicted them. So it means that in sequence terms, experimental and real complexes are the same, and unshuffled are different. But in terms of structure, none of them are similar. Just to be a bit more illustrative, shuffled means we took, let's say, this navy one and combined them with the blue, CM, and all others. But the real one, it's only the navy one and the pink one. I hope it's clear because it's very important for all the rest of the results and findings. Now, moving to our observations. After structure prediction, we just look into the confidence scores that all of the recent structure prediction tools provide. In this case, we looked only at the AlphaFold 3, Boltz 2, and Chai-1 because they were the most representative tools in, let's say, each category, and also they were the most used and talked about. On the y-axis, you can see 106 VHH PDB IDs, and the x-axis, the antigen PDB IDs. So let's say if we believe that structure prediction tools can accurately predict antibody-antigen pairs in terms of the right placement of the paratope and epitope and the right structure of both complex and each component, we would expect the diagonal, full diagonal. Each cell is IPTM score, but let's say it's just in general confidence score that tools provide. But you can see that the diagonal that represent real complexes happen only at the AlphaFold 3 case, but not in Boltz and not in Chai. The darker cells reflect higher confidence of the tool in the prediction, the lighter one, the lower confidence. But in general, you can see that there is no strong consistency in terms of predicting only real complexes and not predicting non-real complexes or shuffle complexes. What does it mean? It means that tools cannot tell you whether the binding actually happens or do not when you do not know it yourself. Then we try to summarize this data in more quantitative terms and more statistical terms. So we gather these heat maps into the violin plots, and you can see a similar trend. That the distributions and the means with standard deviations between in the matrix between real and shuffle complexes overlap quite a lot in all three cases. In terms of AlphaFold 3, as we see here too, it is better in recovering the diagonal, meaning that to understand the difference between real and shuffle complexes, but still there's quite a lot of target prediction. The same in terms of pair out. What's more interesting, when we compared the outliers of each of the structure prediction tools, we found out that they do not overlap in this case. By outliers, I mean, if you look at these heat maps again, let's say chai, there is, let's say, these cells that are dark and have high confidence scores, and we wanted to figure out whether this VHH antigen pairs that outliers with chai scores are also outliers with boards and AlphaFold. And this analysis of overlap of outliers showed that they do not. The correlation between the outliers across tools is very, very low, meaning that those complexes that will be predicted either exceptionally well or exceptionally bad will vary from tool to tool. And you cannot say for sure what to believe. Also, it can tell you that, let's say, if you cannot predict an accurate structure with one tool, you can try another tool, and maybe you will have better luck with that one. Next, we looked into more physics-based scores. Before, I mentioned confidence scores like IPTM. We also looked into PLDT and similar things, but overall, they were AI-based metrics. But this score, called top model score, is based on geometry and structure and calculate the number of clashes and inconsistencies in structure. In other words, if top model score called clash score is high, it means the structure is wrong and has lots of structural and geometric inconsistencies. Interestingly, that you can see that this clash score is quite high for all of the tools because it's, let's say, on average above 20. But interestingly, it's almost double worse for Chai-1 case. But other than this, you can see that for both shuffled and real complexes, the distribution is similar, meaning that the accuracy of the structures for both cognate and non-cognate pairs are relatively the same, and the same not very good. One more comment regarding the three different subplots. We call them train, test, and both. This is just a subset of the complexes that we use either in training set for all the models, only test set in those models, or it was a mix. Moving forward, we wanted to check whether models know when they fail. So we look at the IPTM as an example of a AI-based confidence score and the structural quality metric like DocQ, and plotted them one versus another. So what would we want to see is that if the confidence score is low, the DocQ is also low, because then it will point out that the confidence scores know when the structure prediction quality is also. But you can see that it's not the case. And in some cases, high IPTM correspond to low DocQ. And there is not really a correlation between IPTM and DocQ. This means that you cannot just use, let's say, IPTM and be sure that your structural quality is great if IPTM is great, or vice versa. But DocU is more reliable because it compares to the templated experimental structure, and IPTM is a bit of a black box metric. Next, we looked into how we can actually improve the situation. One of the typical things suggested how to increase the accuracy of the structure prediction is to improve sampling. And by sampling, I mean how many times you predict the same complex. In this case, we tried 1, 10, 25, 50, and 100 predictions. So the amount of samples per complex. And overall, we see is that sampling does improve the structural accuracy, but not the confidence. For example, in case when with just one sample, the complexes had quite low DocU, which is structural accuracy. But when we predicted more and more of them, the DocU became better. However, as I mentioned before, since DocU and the confidence scores do not really correlate, this tells us only that if you care about the structural accuracy, like you want to get the likeliest reasonable structure, it's better to have multiple samples, but you still should not believe in the confidence metric reflecting the binding specificity or any functional properties. Next, we look at the epitope recall. In this point, epitope recall is the number of residues between the predicted complexes and the real complexes that overlap. In other words, it's if you take the binding residues for the experimental structures, how many residues in the predicted complexes will match them? And we plotted the same matrices, the same heat maps as before. So we again expect that if with predictions we have the right binding pairs and the correct structures, we will have the diagonal. But again, you don't really see the diagonal. Maybe you can see a bit of a diagonal for whole 3Ks and both 2Ks this time, but still, it's quite diverse and you don't see the specific trend. What it means? It means that you really need to be careful with the structure prediction, especially when you don't know when and where your proteins bind. Because just based on the confidence score, you cannot say whether the binding site is correct or not. So ideally, if you can add any information from the literature, experimental information about any ideas where the binding happens, this can improve your structure prediction a lot and confidence structure prediction a lot, especially taking into account that some of the models allow you to do restraints by guiding to the right binding site. But by default, you need to be really careful in predicting complexes where you don't know where things bind. Similarly, we try to use multiple samples and replicas to check how the epitope recall will change with more samples. And we found very similar things to the previous observation that with more samples, the probability of finding the right binding increases, but increases just relatively, because you still cannot say for sure when it's the right or wrong without experimental structures or any other like literature or experimental information. But also it's quite soon reaches saturation without improving the specificity. What does it mean? It means that if you have the opportunity to do replicas, you should do replicas, meaning that's sample at least 10 times per complex. This way you can get better structures in terms of the accuracy and the quality of the structure itself in the complex. But it will not point you to specificity. Also, it tells you that you don't need to do sampling to infinite. You don't need to do thousands of samples or even hundreds, because the improvement with each next sample after, let's say, 50 will not be that big. And maybe you can use those computational resources for a better and more thorough biophysical analysis to really understand whether your predictions are good or not. Next, we look how structural information correlate with epitope recovery and epitope recall. We looked at the structural consistency across the samples. So we used that multiple samples and calculated the mean pairwise RMSD across replicas, meaning that we just align all the structure pairwise in the sample and looked whether they're different or similar. And in this extreme examples, you can see that in some cases, the samples are very similar. For example, here you can see that there are actually 10 samples here and one here. So in majority samples, the binding site is the same with one outlier. But in other cases, the binding site in all cases is the same, relatively the same, but the orientation is a bit shuffled and a bit on different angle. And the same here. What does it mean? That this is exactly the examples of improvement with the sampling. If you have just one sample per prediction, you might end up in this situation when you have the antigen and the wrong binding site. But if you will repeat samples, let's say 10 times, you will have 9 structures here and 1 structure here. So it's likely that this is the right binding site compared to this, just because here there is 9 predictions that end up here and here it's only 1. Of course, it's an heuristic, but this is something that you can use for your internal checks if you don't have anything better. Importantly, we actually tried this hypothesis of RMSD connected to the better estimation of prediction on the whole data set. And you can see a slight trend that in real complexes, RMSD between samples is lower compared to the shuffle complexes. It means that if you are dealing with the real binders, likely you will have something like this, where all your samples will be very similar in terms of structure and positioning of the binding. And if you work with non-binders, you will get something like this, and maybe more extreme case, where you have, let's say, here one binding site, here's one binding site, here one binding site, etc. And you can use this to test and supply this to your own observations. For example, if you do not know whether the antibody-antigen pair is a binder or non-binder, you can, let's say, generate 10 samples or maybe 20 samples for a complex and compare it with the 10 or 20 samples for the complex where you know the binding. Let's say you calculate RMSD between the samples in your complex of interest and known one, and if they're approximately the same, you can think that might be likelier that the complex is a binder. But again, it's more of an heuristic and works better when you work in high throughput manner rather than really, for sure metric of differentiating binders from non-binders just using in silico metrics and computational tools. Next, we actually looked into energy consumption because it's something I personally think is quite overlooked. Because let's say in many papers you can see that, let's say, sampling improves the structural accuracy of the structure predictions, right? But then sometimes you can see that recommendation is to do thousands of samples, but it can be far, far, far more than many can afford. And also, it can require so much computational resource that it might not make sense to do that. So that's why we actually check the energy consumption. And overall, you can see that first, it might not be that surprising that energy use varies by tool. And the next thing that even though that sampling improves the best case scenario, you still reach this plateau, which is similar to one of the previous findings where I mentioned that be smart with the numbers of samples you're using. It's a good idea to use several samples, and several depends, of course, on your computational resources and capabilities. But let's say the rule of thumb 10, 50, from 10 to 50, it's something reasonable and can work well for you. But above that might lead just to incremental accuracy and plateau effect. So please do sampling, but you don't need to do too much sampling to get the same results. Another thing, what we found out is that actually the largest improvement in accuracy and the quality of the structure prediction happen when you just start sampling. For example, it's also what I mentioned before about the plateau effect. It is significantly better to have 10 samples compared to 1, but it's already less significant to have 50 samples compared to 25. So the more samples you have, the lower the improvement. And similar to our quality of the structure study, the sampling helps to remove the incorrect predictions by for example, highlighting the situations. In statistical terms, you will see that with more samples, if some conformation somebody stays stuck, for majority of samples, this is a good indicator to believe more into this complex. Of course, if you don't have any information that says otherwise. And one of the final findings was the importance of seed selection. The fun fact actually is that seed does affect the quality of the prediction, likely because of such cases where you can just accidentally with one sample pick the wrong conformation and predict the wrong conformation and the binding site. But again, if you do even 10, 20 samples, the effect of the seed will be reduced because you will saturate the differences in the structure prediction. It was quite a lot of information. I summarized some of the main conclusions on this slide, but you can also check out the paper or reach out to me later if you have more specific questions. We are working on the revision right now, but all your input and any suggestions on how do you think things can be more clear or more properly proven, we are all welcome to hear them. And thank you very much for your time and listening to my talk.
Thank you very much, Eva. That's very impressive and super interesting. I think we'll have a lot of questions, but let me start. And please, everyone who wants to ask, you can just drop in the chat or use a QA button in the meeting panel.
Yeah, so, I see right now one question actually in the chat. Thank you. And did you plan to try IPSA? Yes. So we are right now adding it to the paper. I don't have the data for all of the tools, but I think I have it for Chai and bots right now, and I think the results were pretty the same. Also, apart from IPSA, we looked into PLDT, an aggregate score, and PTM, and they were also quite similar to the IPTM findings. But specific results for IPSA you will probably find in the revised version of the paper quite soon.
Yeah, meanwhile, another question. Okay.
Yeah, on IPTM versus DocQ, can the interface be the same, but the structure may be mobile so that DocQ gives low score? Yes, it can be, but as I mentioned before, we try to get very different structures of VHHs and antigens. In this sense, what I mean is that quite some of them are quite rigid, meaning that the impact of mobility, especially on the antigen side, might not be reflected that much in this case.
Yeah, well, thank you, Eva. And let's continue with the questions then. We have one question in the QA from Satya Murthy. The question is, from isomorphic labs, accurate predictions of novel biomolecular interactions with iso-DDE. Is it solving the problem? That's the question.
Yes, so I would say that it's there are several answers. First, it's a commercial entity, right? You will not have access to that tool. And also, it is not really clear what the architecture, what was made to reach such a drastic improvement compared to AlphaFold 3. So in this sense, it's very hard to say how much the improvement is in, non-traditional benchmarking and maybe in a more real-world setting. And I believe that to really say solve this problem, we need to have a tool that other people can use freely and also easily to test and understand what is actually going on inside, because it's very hard to make conclusions when you just have the final numbers and you don't know how things actually work. So it is my personal opinion. But let's say if we just look at their numbers, they did improve. I don't remember how much, but like 4 times, I think, compared to AlphaFold 3. But 4 times compared to AlphaFold 3, the baseline was not very good, right? It's like, let's say if you have 0 as a baseline and then you improve it 4 times, it's also 0. If you have 1 as a baseline and improve it 4 times, it will be 4. If you have 20 as a baseline and more is 4 times, then it will be 80. So I think it really depends on what you compare it to and what is exactly solving the problem. I think, again, I would really want to see how it really works on non-typical benchmark datasets and a real-world example to be really, claiming solving any problems or revolutionizing drug design.
Well, this actually brings one of my questions right here. That's marketing versus reality. There is a real gap between the science and the papers like yours, showing what those models can do and can do, and the announcements from the companies like Isomorphic Labs, Chai Discovery, and others, which sound like the problem is solved already, almost. Yeah, we are almost there. So how do you reconcile those two narratives? Do you see that the field on the commercial side is just overselling, or are they actually like solving maybe a different problem than the one that you're showing in the paper?
I would say it's really, really, really hard to find what is it true. And I would not say that, that like startups and pharma are the evils and academic labs are saints. I think in the current world, marketing is everywhere, and you really need to be careful in what to believe. And I personally try to less pay attention to big announcements and big titles. Let's say I try to not really focus on whether something is revolutionized the field, but whether is there some practical and usable improvement that I can use or the field can use. So I really try to just either try or look into the things in a very objective way and try to look into how the things work rather than just reading the paper without trying to apply it. Because I think there is a huge gap between, let's say, even the best theoretical or like on the paper thing might not work in the practical setting. So I would answer this way.
Yeah, well, thank you, Eva. And right, since we are here on this slide, which impressed me most when I just started to read the paper, yeah, where we see where like Boltz 2 is sure about everything. Chai-1 is unsure about almost everything, and AlphaFold is somewhere in between. So the results are here on this plot. But do you think this, let's be honest, failure of those models to predict the thing is the fundamental restriction of how those models are built? Yeah, or it's just, we will improve, include more training, and it will become better.
Yes, I'm a bit old school in this sense. I came to the field when there were before AI and when there was this physics-based methods. So I really think that having a bit more interpretation and explainability of the models and the parameters can help. So answering your question specifically, none tool is perfect, right? You can always improve it. And whether it's the issue with the tool per se or the approach, I think there have been lots of tools tried out, right? I think the thing is that people try to build on what already has been proven successful. It's a bit connected to your previous question about this question, the statement that something revolutionized the field. AlphaFold did a great progress, right? But then more and more people start to use AlphaFold real architecture instead of trying to invent something new, because AlphaFold already proven that it works. And in this sense, it's like if we all make mistakes, we all do the mistakes into one direction instead of trying different mistakes. And I would say that it's less about the tool and architecture, it's more about trying really diverse things and be less punished by trying different things, especially if they're wrong, because then you can see that, okay, that did not work, but at least you know that another direction does not work. But you can try a very different direction again, and then it will be the next AlphaFold tree. Right now, I feel like people a bit more shy and a bit more trying to pick the better small win than the big lose strategy. And in this way, it's really hard to answer questions like you ask, whether it's, let's say, the issue with the model or you need more training data because something drastically different was not really published or tried properly. I personally think that just adding more training data is not sustainable because it's like, again, you need more. It's a lot. And okay, you will add this more data, but then you just get overwhelmed with it because you will not be able to understand. And there will be only AI models that are able to work with. I personally think that if you have something like physics laws that explain how things behave, you don't always need more data. You need more data when you don't understand things. So there's at least two ways. One way is to find, of course, get more data, but I think there's already lots of data. So it's a question of right processing, finding the right features to really make this next step. But another way is to actually try to not add more, but finally figure out what's going on with what we have, because I do think we have already lots of data, lots of models, and figure out to try to make biology a bit more like physics and establish these laws that will be able to be reproduced rather than just learned.
Well, thank you very much, Eva, for this rigorous answer. We have a lot of other questions, but before them, I want to follow up on this a bit more because it's really interesting for me. Like when you're saying physics, I really love this because I'm a physicist by training and I was doing physics a lot. And my favorite company is Schrödinger. But we all know that it takes hours or days for like physics-based modeling. And talking about like new ways and new models, new approaches for things like that with AI requires a lot of compute. Like you run a lot of simulations. You need access to, I don't know, computers, GPUs, etc., which are now concentrated more and more. Well, and you have to pay for them. And what you're saying sounds like this new discovery and new approaches can be done only kind of in academic settings where you do research. So do you see a disconnect between what's accessible, what is accessible in terms of, compute power in academia, and whether it's enough to do progress in that field?
I would say it's usually very polar. I would say few have lots of resources and most have very little resources. And those who have very little resources need to come up with creative solutions, I would say. But honestly, I don't think that it's always a bad case, because in many situations, creative solutions can be better solutions. Because as I mentioned before, adding no training data, let's say it's a simple way, because people already see the trend. More data, more resources, it's easier to just scale up. But whether it solves the problem, it's already a different question. And another thing is, like, I'm really, to be honest, inspired by the work of lots of smaller labs, smaller companies that are using less resources, but jumping way over their heads in terms of providing the results. And I think this is exactly the smart ways to move forward, rather than just scale up things that try to really find the problem and solve it, and not, leverage the muscles. And whether it's only in academic setting or pharma and companies, I would say that there are some startups that has lots of funding. There are some startups that have very little funding. There is also some labs that have lots of funding. There are some labs that have very little funding. It's a bit controversial statement, but I think that you might not always need as much funding if you know what you're doing, because you can really test. If you have some specific ideas and some specific solution that you already have in mind, you don't need to probe around, so you don't need that much funding to probe around, and you can just use that funding to test just that exact solution and answer. It might be wrong, but at least you will not use most of your funding just to explore around. And in this sense, maybe something average, like, of course, not completely zero resources, but some resources will be a sufficient middle ground in this sense. And for this, well, like academic labs, especially with computational resources, now it's getting a bit easier with Google Colab and some discounts for academic labs. Let's say even NVIDIA has discounts for the simulations. For startups, it's a bit harder, but I would say that there is quite a big shift in funding in general towards the computational biology field. So I think it might be like higher competition, but also a bit more funding in this area. But honestly, yes, simulations take a lot of resources. AI model development also take a lot of resources, but there are still some things that you can test if you have very specific questions and solution in mind, even, let's say, in your own laptop sometimes, or like with Google Colab. And then these answers for specific questions and specific solutions can give you the next finding that you can already be use more exploratively and for bigger simulations and similar things.
Thank you, Eva. I'm now switching to the questions, and there was a question from Garrett Morgan. Were any of the structures known to be in the tool's training sets, and were the predictions better for these structures? Were the predictions better for these structures?
Yeah, we know for all of the structures whether they're trained, test, or both. Both as are cases with the shuffle complexes, because it's like, let's say, either antibody from the train and antigen from the test, or vice versa. And the second part of the question, whether we know how it behaves specifically for specific complexes, or what was the second part of the question?
Yeah, were the predictions better for these structures that are known were included in the tools training sets?
Yeah, I mean, for example, here you can see the three columns, right? You don't really see much difference between train and test. The same here, the same here. And I would say that similar findings are for all the parameters that we looked, which does not look good, because it also shows that on a simpler task, when the model already saw the trained data, it still makes mistakes in antibody-antigen complexes. Partially, it can be because antibody structures are very underrepresented in the training for those models in general, but also it can be because of the unique interfaces in the binding structure of the antibodies and VHHs, right? Because they have this flexible loops. It's a bit connected to the question of Mikhail about the mobile interfaces. So maybe conformational variability and mobility is something that also make antibody structure prediction that hard.
Thank you. Another question we have from anonymous attendee. Can you share your experience with T-Fold from Tencent Labs? It did not make a big commercial splash, but the end-to-end workflow shows promising data compared to AlphaFold 3, etc.
I have not used T-Fold. Is it similar to AlphaFold or Rosetta or any other tools? Because I mean, there's also Platonics and many other structure prediction tools, right? But this T-Fold, does it use a completely different architecture and approach, or is it similar to, let's say, AlphaFold? Because if it's similar to AlphaFold, I would probably expect that it will become the same, but I myself did not test it, and I don't.
Thank you. I also did not test. I don't know this answer, but if the one who asked can follow up in the chat, then we will have that. So another question from Jeremy Sheck. Given the limitations you've shown, what would your recommended workflow look like for predicting an antibody-antigen complex currently?
Yes, so the first question is what kind of a priori information do you have? Let's say, do you need to predict just one complex and you know nothing about it? Or do you need to do high throughput structure prediction? And you know, let's say that there is the same binding site that all of this variants share. So first step that I would do is to clearly define the question and the study setup. Whether it's one or many complexes, whether you know anything about the complexes, whether you have any similar complexes, or in terms of both sequence structure or maybe biological function that you can use as a priori information. Also, of course, literature and experimental. But let's say you know nothing, and you just need to predict the complex where it binds. And let's say you might also not know whether it binds at all. In this way, then I would use several tools, likely this AlphaFold 3 bolts 2 and Chai, or whatever new tool will appear and will say that they're better. Then I would use multiple samples. I would, as I mentioned, use like 10 at least samples. Of course, depends on how much resources I have. But let's say I would use 3 tools, 10 samples per each tool. And then I would visualize them first and align. If I see something like this signals me that likely if they bind at all, this is a likelier binding sign compared to this. But if I see something like here's one, here's the second one, third, so VHHs are spread all over the antigen surface, it would be a red flag. In terms of first, the model does not know where exactly it binds, and second, it might be a signal toward that they're not binders at all. The next thing, I would calculate RMSD between the samples. And I also would do the structure prediction of a complex that I know for sure is a binder. And also I know for sure that non-binder. For binders, you can find this information like in PDB, for example. And for non-binders, we're actually right now doing experiments to confirm specific non-binding pairs, so you can use this information too. I would use that information for binders and non-binders in terms of RMSD as baselines. So I will have, let's say, mean RMSD and STD for binder, proven binder, and proven non-binder. And for non-binder, we will have higher RMSD, as this observation show, and for binder, we will have lower RMSD between samples. And then I would put my complex right now into the scale. Let's say if this new complex will be somewhere here, then I would presume that they're non-binders. But if they will be somewhere here, then I would say that they're binders, and the detected by majority binding site is likely be the right one. And especially if this observation will be consistent across different tools, then it will be like +1, +1, +1 signal towards the confidence in my assessment. Is it clear?
For me, yes, more or less. For the one who asked the question, I hope so. So, we'll see. We'll see. Meanwhile, we have yes. Thank you. Yes. So
Perfect.
meanwhile, some more questions from the chat. So Maria is asking, hello, thank you for an amazing talk. What do you think about antibody-specific structure prediction tools? Does AlphaFold 3 outperform them significantly?
Yes. So I tested Antibody Builder, let's say, and IgFold, but it was like 2 years back. So those times they did outperform. But right now, I would say I don't know. And I would say that I would use those tools that are better maintained and also have the updated versions more frequent. Because let's say IgFold was great, but it's not as frequently updated or maintained as AlphaFold or Bolt, for example. And when it comes to tools, and especially their longevity, it all comes down for the maintenance and updates. Because with each new update, you will get a better prediction and better score. I would also be careful with over-reliance on specific to antibodies tools just because they might have artifacts and issues in just general structure prediction and representation of the structure because of the bias towards antibodies. Like they did not, they might not know or might have forgotten the just general geometric and like structural rules in terms of how to build the structure. So it might be a good idea to check the antibody-specific structures, but let's say if it comes to the practical setting, I would, let's say, use maybe one antibody-specific tools and two specific general tools, or maybe like proportion of two antibody-specific to general. And again, I would use those tools that are really well-maintained and have updates more frequently just because it's practically better and long term it's also more reliable. Also, one more thing, I would be careful with, you know, using tools just based on publications because one thing is to publish a paper, another thing is to use the tool. And installation, running with all the parameters, usually, more general and bigger models are better in these terms. So even the advantages that some antibody-specific tools have, they might not work fully just because it will not be you will not be able to install it on your own computer.
That's so true. So true. I can tell you. So we are doing in our company those like packaging of tools to, democratize access to those things, and it's very hard. Well, thank you very much for this answer. We have few more questions, and we have few more minutes. So, question from Satya Murthy. What is IPTM cutoff in Boltz 2 given accurate prediction?
It was above 80 or like, well, actually, if you look here, I don't know if you can see my screen very well. I'm not sure that I can zoom in, but let's say if you see my mouse, this is 0.9. And I'm pretty sure that most of the cells here are 0.9. So in my specific example, just having the cutoff more than 0.9 wouldn't be sufficient enough to make this differentiation. So yeah, I mean, here it's less of a specific cutoff to figure out whether the tool gives the accurate prediction and more about whether the tool can give an accurate prediction at all at the setting. And I think in this case, it's like, even if you put it very, very high, it will not be differentiative enough. And if it's more of a general question, we all have this PLDT cutoffs, IPTM cutoffs, etc. But I think it's more of a question whether these cutoffs work for a specific your system and your task or not, because they might not work all the time.
Thank you, Eva. And the last question for today from Andrew Aiginin. Hello, thank you for a great talk. It looks like we still don't have a strong oracle model that can score binding poses and identify the best ones. Do you have any ideas on how an oracle model that discriminates between real and plausible poses could be trained? What kinds of parameters could be used to train such a model?
Yeah, it's an excellent question, and you're totally right. We don't have an oracle that is able to tell us for sure whether there are functional and real like biological candidates or not. So first question about let's say training. One good idea to train on with the negative data too. For example, explicitly tell the model and shows the model when the specific sequences and ideally also those specific structures are non-binders. Because I think that most models do not know that there is such a concept as non-binder at all, and they just try to always assign, some binding answer. So train a model so it understands and knows that there is also non-binder option. That's a good idea. And for example, our data can be something because it contains these non-binders, but also, maybe try to generate artificial non-binders by, for example, putting the structures, antibody-antigen structure far away from each other, or maybe, creating dummy sequences like, the whole alanine, just alanines, and I don't know, instead of CDR of the VHH and the antigen, and then of course you expect it will not bind, but just to create this negative data, either from the real experiments or from synthetic data or from PDB or from whatever resources of creativity you can find, and to teach and show model that there are such things as non-binders and negative data. And regarding the parameters and features, I honestly think that showing and having a bit more explainable and biophysical parameters would help, just because then you can trace back the mistakes of the models. Because let's say if it's like layers after layers after layers of some black box, it's very hard to understand where exactly the mistake is made. And having some trackable things like biochemistry interactions, this is easier to track and to understand when things goes wrong. So I would look into parameters more by physical parameters, like let's say just hydrogen bonds, different types of bonds. Also, how much the residues are exposed or not, and try to incorporate them into model. But the first thing is probably again to train on this data with specific negatives, and then see whether it already can actually separate or not. Another thing that might be missing is flexibility information, especially for antibodies and their like CDRs that are changing conformation upon binding. This is something also you can take a look into. Like an
Thank
examples.
you. Thank you very much, Eva. We have more questions from Mikhail and Sophia. I'm sorry, Misha, Sonia, but we have to wrap up. It's time. I also was able to ask only 10% of my questions because this topic is so interesting. There are so many insights you shared with us, Eva. So massive thanks. Please, who has questions, just feel free to follow up, either me or Eva, and well, so you will
Yes.
have the answer.